Two plants, two worlds, one microscopic truth. Beauty and defense are not opposites. Nature hides both in ways our eyes were never designed to see. The morning glory, known scientifically as Ipomia purpurea, reveals impressive structural complexity under microscopic examination. Its petals consist of thin parenchymal tissues interwoven with pigment-rich cells that contain anthocyanins. These pigments produce the flower's blue, purple, and magenta hues. Because anthocyanins are pH-sensitive, the coloration intensity changes throughout the day.
At the center of the flower lies the corolla tube, which houses the stamens and pistil. The pollen grains of morning glory are generally spherical, with a fine reticulate pattern on their surface, providing increased adhesion to the bodies of pollinators. Along the inner lining of the corolla tube are nectar-producing cells that secrete a sugar-rich fluid essential for bees, butterflies, and other pollinators.
The heart-shaped leaves of the morning glory have a relatively thin cuticle and a high density of stomata. This results in significant transpiration. Microscopic hair-like structures on the leaf surface help reduce direct water loss and provide limited protection against small herbivores. In contrast, the pumpkin plant, Cucurbita pepo, exhibits a very different anatomical design. Its leaves are large, broad, and angular. Under magnification, the leaf surface is covered with thick, coarse trichomes. These hair-like structures serve as mechanical defenses, making it difficult for herbivorous insects to feed on the leaf tissue. In some species, the trichomes can even contain chemical deterrents. The pumpkin leaf possesses a thicker cuticle, offering enhanced protection against intense sunlight and water loss. Its stomata are often recessed within grooves, reducing unnecessary evaporation. Internally, the leaf contains robust veins with well-developed xylem and phloem, supporting the heavy nutrient demands of its large fruits. Under microscopic examination, the pumpkin leaf reveals a hidden ecosystem dominated by colonies of aphids, tiny soft-bodied insects that feed by inserting their needle-like stylets deep into the plant's phloem. Each aphid is equipped with a pair of cornicles on its abdomen, small tubular structures that can release defensive droplets when the colony is disturbed. Their bodies are translucent, allowing internal fluids and digestive motion to be faintly visible under magnification.
Aphids reproduce with astonishing speed, often giving birth to live clones without mating, a process known as parthenogenesis. This allows a single female to generate an expanding population in just a few days.
As they feed, aphids excrete a sugary substance called honeydew, which accumulates as shiny droplets on the leaf surface. These droplets attract ants, fungal spores, and other microorganisms, turning the leaf into a dynamic, multi-species microhabitat.
Under high magnification, the interaction between aphids and leaf structures becomes more apparent. Trichomes appear as towering pillars, and the aphids navigate through them like a forest.
Their mouthparts penetrate between epidermal cells with remarkable precision, enabling continuous extraction of sap without causing immediate tissue collapse. This constant feeding can weaken the plant, reduce photosynthesis efficiency, and create entry points for pathogenic microbes. In this hidden microscopic world, every aphid plays a role, both as a feeder and as a potential target for predatory mites, parasitic wasps, and other microscopic hunters.
Overall, the morning glory represents a fast-growing, delicate anatomical design, while the pumpkin plant embodies strength, resistance, and structural efficiency. Both plants, though fundamentally different, demonstrate highly optimized biological engineering. And I know that God is nearby. From the microscopic pigments of the morning glory to the defensive trichomes of the pumpkin leaf, every detail reflects the precision and intention woven into the fabric of creation.